talk about yet flying is on other receptor. How the unmasked protein are, how the proteins are unmasked?
They help the clustering
How the proteins are unmasked? That's my question.
by albumin.
Okay.
Albumin unmasks the proteins as well as the cholesterol, so it helps the clustering of the RAS.
Good.
And then albumin can also extract carbohydrates and other proteins to unmask the receptors. And then all the proteins that are exposed on the sperm head will help bind to the receptors.
Okay, so now we want to talk about this, the mechanism of capitation, how the capitation occurs. And we said there are two types of events which are occurring. And those events are occurring at head and tail. The events which are occurring at the head, they are going to prepare for acrosomal reaction, and the events which are occurring at tail are going to provide hyperactivity. That means the flagella will start moving like this. Okay, so what is happening there? So think about, this is the membrane of the sperm, this is the membrane of the tail. Okay, so we'll talk with the tail first and then we'll go to the head. So events occurring at the tail. So we are talking about the tail first. So what is happening at the tail? We are saying that this is the membrane on the tail side, right? And what happened that the albumin has removed, so this is the cholesterol removed from tail membrane. So cholesterol is removed by the albumin. And when that is going to happen, two things are going to happen simultaneously. Right here, so this is the membrane. Right here on the membrane, you are going to have, these are called bicarbonate channels. So bicarbonate channels are there. And what is going to happen, this removal of cholesterol by the albumin is going to activate, let's say step first. Step first, activate bicarbonate channels. Okay. And when the bicarbonate channels are going to open, from here, the bicarbonate ion are going to get in. So bicarbonate ion are going to get in the cell. Okay. Now on the other side, what is going to happen? Removal of albumin and opening of bicarbonate channel together, they are going to influence right here, there are the other channels right here. These are potassium channels. Potassium channels, they open. So when the potassium channel open, where potassium will move? You can talk to each other and give me the answer. Give me the right answer. You can talk to each other, give me the right answer.
This is actually a negative ion. Can you write that? It's CO3 negative.
Okay, where will they move? Where is potassium concentration high? Inside the cell or outside the cell? Usually in our cells.
Inside.
Inside the cell. So where they should go? Is different. They should just go out, right? So that is going to lead. So this is the step 1, step 2, potassium channel open. And when the potassium channel open, what is going to happen? The potassium ions, they are starting to move out. So potassium ions start moving out. Now, these two events, bicarbonate ion coming in, what it is doing, it is increasing alkalinity of cytosol of sperm, right? And then what is happening there, potassium going out is going to make these channels inside more positive or negative. Membrane will become more positive or negative. It will become negative because positive charge is going out. So that means there is voltage change, right? So on the cell membrane, right here, these are voltage gated calcium channels. Voltage gated calcium channels are there. And these voltage gated calcium channels, they will open, but they open only if there is high alkalinity. Okay? So, two things are happening which are allowing these channels to open. The change in voltage and change in alkalinity, right? So, effect of these two things is this third event which is going to happen, that the voltage gated potassium channel open. That's your third event which is happening. And when that happens, that leads to movement of calcium from outside to inside. Calcium moves in. So far, it is good. Now, this calcium which is right there, we are still on the membrane. Put this thing a little bit up, you know what was that. So we are still on the membrane. Here is an enzyme, membrane-bound enzyme here. This enzyme is called sperm adenylyl cyclase. Sperm adenylyl cyclase, also called SASI. I'll write here SASI. Okay. So this is an enzyme. This enzyme is activated because of two reasons. The bicarbonate ions and the calcium ion both go there and activate SASI. Okay? When that sperm adenylyl cyclase is open, what it does, it converts ATP to cyclic AMP. And cyclic AMP activates protein kinase A, protein kinase A is activated. Okay. And then when the protein kinase is activated, that phosphorylate tyrosine kinase. And once this tyrosine kinase is phosphorylated, this is actually activated. So that is activated. And once that is activated, that phosphorylate various sperm proteins. So that kind of, so I'm putting this arrow right here. Okay? Now remember, we had a lot of calcium here. If you remember from sea urchin, what was the role of calcium early in the movement? When the sea urchin gained the capability to move and go toward the reset, what it does, what calcium does. Calcium, right here, activates dynein, right? Dynein is a motor protein there. If that is activated, that is going to make flesh-love movement. And dynein utilizes ATP, it's a ATPase, right? And we said from where ATP is coming, from the mitochondria. And we also know what calcium does. Calcium activate mitochondrial ADP production. Okay. Now, various proteins which are phosphorylated along with, I'll show right here, along with all this thing which is happening, what that does, that activates sperm. And now sperm becomes hyperactive. And that becomes so hyperactive. Now this sperm is attached to isthmus, right? So have you ever noticed a little kid? You hold the hand of the kid and show him candy from there. You know how difficult it is to control that kid? The same thing happens here. So that sperm which was whole, now this has become like very hyperactive. And then the sperm which are hyperactive, it means they gain the capacity to move to the egg. I didn't talk about the direction yet, but they gain the capacity, right? So they are removed. So this is one part of the capacitation which is happening at the tail. So they become really hyperactive. So let me go back so that what is happening. We are saying that in the sperm membrane, we are talking about the tail first. We are saying in the tail, the albumin removes cholesterol. And that removal immediately causes two events. The first event is opening of bicarbonate channels. Bicarbonate ions are moved in the interstitial fluid. Remember, why they are produced? Because they are produced because carbon dioxide come out of the tissue, combine that, make it carbonic acid. Right? So they are too high outside the cell. And then the bicarbonate ion gets into the cell. When the bicarbonate ion gets in the cell, when this open, immediately along with this potassium channel also, and potassium start going out. Now that is decreasing the voltage inside the membrane and also making it more alkaline. Increase in alkalinity and change in voltage cause opening of voltage gated calcium channel. Calcium is always high outside, that starts running inside the cell. Now when calcium runs inside the cell, this high level of calcium along with bicarbonate ion, they activate sperm adenylyl cyclase, which is called SASE. Once SASE is activated, that converts ATP to cyclic AMP. Cyclic AMP activate protein kinase A. Protein kinase A, say kinase, add phosphate group to tyrosine kinase, and tyrosine kinase is phosphorylated. So once tyrosine kinase is phosphorylated, it's become activated, it phosphorylates other sperm protein, kind of activating all the sperm tail proteins, enzymes, so they can work. All those enzymes, along with increase in calcium was there, that activate dynein, activate mitochondrial ATP production, and that caused the sperm to become hyperactive. Now sperm flesh will start moving like crazy. So it's kind of like that fast movement does not allow the isthmus to hold it anymore, and it's removed from there. That is okay. So talk to each other for a second, then we'll go further. What is happening in the head?
Sorry. The sperm's like this. No, that makes sense. Okay, any doubt here? Okay, now what is happening at the head? Almost the same events. So what was the first event there? First event there? In the tail, what happened first? Opening of? Opening of bicarbonate channel, right? Then what happened? Opening of? No. Read, look at your notes and then tell me. With bicarbonate, what was open along with that? Before voltage gated calcium channel. Potassium channel. There was no voltage gated. Voltage gated channel or calcium channel. Opening of potassium channels. What this cause? Bicarbonate ion entry, potassium exit, right? So these two things, what they do, both of these together. Increase the alkalinity and open the, so increase alkalinity and together this open voltage gated calcium channel, right? It's happening like that. And then what happens? What calcium does and bicarbonate ion do? They activate SASI, right? So bicarbonate ion, SCO3, and calcium together activate this. Okay, once that is activated, we are saying we have ADP changes to cyclic AMP, right? And we are saying cyclic AMP activate protein kinase A. This is common, right? You have a lot of calcium channel, you have protein kinase A activity there. Now, after this, what happened? We have few things here. We have high calcium. We have high calcium ion. Then we have high cyclic AMP. All these are high cyclic AMP. And we also have higher amount of bicarbonate ion. Right? What these three things do, these three things together, even only calcium is enough, but when these three things are together, what they do, they facilitate membrane fusion. Okay. The other thing happened when this protein kinase A is activated, it phosphorylate a protein, and that protein name is isomo. Okay. When this protein is phosphorylated, this is present in the cytosol, right? But when this protein is phosphorylated, isomo moves to acrosomal vesicle membrane. So what is happening here? Let me draw it here. Just the head, okay? So this is sperm head membrane. Here all those things have happened, all these things are happening here, right? So because of high calcium, high cyclic AMP, high bicarbonate ion, this is the acrosome vesicle. This acrosome vesicle is going to move toward this, and the membrane fusion can take place, and that can give you the acrosomal reaction, right? And the second thing we said, the protein isomo, which was somewhere here, what that does, that is phosphorylated, this is isomo, and this isomo is phosphorylated, that move from here to on these sides. It also moves in the membrane. This protein, isomo, is critical. Isomo is critical for sperm membrane binding to egg membrane. So what is happening? We are saying almost the same events which occurred in the tail are happening here. Albumin remove cholesterol, that activate bicarbonate channel, bicarbonate gets in, activate potassium channel, potassium gets out. These together increase the alkalinity and change the voltage, and then that leads to opening of voltage gated calcium channel. Calcium level goes high, cyclic AMP level are going high, bicarbonate level are going high. These together are facilitating the vesicular movement and fusion of acrosome vesicle membrane to the sperm membrane, so that acrosome reaction can take place. And then we are saying what is happening, because bicarbonate and calcium, they are activating SASE that cause cyclic AMP generation, that activate protein kinase A. Protein kinase A phosphorylate isomer. Phosphorylate isomer move from the cytosol to the vesicle of to the membrane of the acrosome vesicle. Now this protein isomer is critical for binding of sperm membrane to the egg membrane. Making sense? So just go over it once and then we'll go further. Okay, any doubt here? Okay, then what happens? Now, the sperm has become hyperactive and it is now ready, because when the calcium is getting in, two events are happening side by side, right? In one event, you are removing it from the isthmus, and in the other event, you are getting it ready for the acrosome reaction, which can happen anytime, right? So now, where to go? So where to go, that's the problem. Now it's become hyperactive, right? So what happened, that in this case, the heat is the Q. Heat is the clue, or Q, whatever you want to say. Okay? So what is happening? The capacitated sperm, now the sperm is capacitated. Once it's gone there, it's capacitated, right? It is actually highly sensitive to heat. It can sense a difference of 0.14 degrees centigrade over a millimeter. Can sense temperature differences of 0.1 degree. So what happens? So now it is following the temperature. And actually, where the fertilization takes place? Where does it take place? Ampulla. Right? So fertilization takes place in ampulla. And where was the sperm now? When it's capacitated? It was in the isthmus. Now, so isthmus is here, it has to reach here. So the ampulla is 2 degrees centigrade warmer than isthmus. So where will sperm go? It's moving from cold to warmer temperature. So then sperm will move from, so therefore sperm moves from isthmus to ampulla. Now sperm is in the ampulla. Okay? Now, So far it is okay? So now the sperm is in the ampulla, but reaching ampulla does not serve the purpose. Okay, you can stay there, right? So the question is, where is the egg? Where is the egg? And that mechanism, what was the mechanism in sea urchin? How the sperm was sensing? Reset, right? So it was releasing something. So in this way, that there should be the similar mechanism here. And we said the first sensing was temperature, right? And the second sensing here is chemotaxis. Okay. And what happened that now the egg is there. Remember, this is the egg. This is your zona pellucida, right? This is your egg, and the cells here are what you call these cells? These are cumulus cells, right? So these cumulus cells secrete progesterone. So they secrete progesterone. Now this is interesting. Progesterone can attract capacitated sperms. Now how it can attract? What it does is, remember we were talking about the calcium channel there. We said that the movement is because of the calcium channel opening. And we said those are voltage gated calcium channel. The same calcium channel which were there, so progesterone can bind to calcium channels. Now, an open calcium channel leading to entry of more calcium. But here is the trick or the caveat. This progesterone can open calcium channel only if high bicarbonate ions are present. Now, high bicarbonate ions will be present only in the capacitated or uncapacitated sperm. Only in the capacitated sperm, right? So, therefore, only capacitated sperms will respond. Now you have more calcium. More calcium, there is more ATP, more dynein activation. And remember, when we were talking about the sperm, we said sperm become really highly hyperactive near the vicinity of oocyte, right? So therefore, highly hyperactive sperm in vicinity of oocyte. That's why even if the sperm reaches within 30 minutes, it's not going to do anything because it's not capacitated. If it's not capacitated, that means it's not going to participate in these events which are triggered by progesterone. Okay? So now, It is now becoming hyperactive, very hyperactive. Now, this hyperactivation is important because now it needs to cross these cumulus cells, right? So the sperm is now, for example, these have reached them, and the sperm has reached here. Now it needs to cross here and come here. First, it needs to interact with zona pellucida. So now, how that is going to happen? So the cumulus cells, what they do now, they were secreting progesterone to make it hyperactive, right? Now the cumulus cell, they secrete CRISP-1. CRISP-1. CRISP-1 is secreted by the cells which are right here. Okay? Now, more CRISP-1 act as a chemoattractant. So, CRISP-1 acts as chemoattractant in hyperactive sperm. This is also important. In hyperactive sperms. So this is the other thing, that uncapacitated sperm, they cannot respond even to this, what we call is CRISP-1. Now CRISP-1 is secreted here. That means you have high CRISP-1 here, less CRISP-1 here. So this sperm, because it is hyperactive, attracted by this, move from here to here to the zona pellucida. Making sense? So we are saying there are three things which are happening. Three things which are happening, which are telling the sperm where to go. So once the sperm is capacitated, it has high bicarbonate ion, it is capacitated, it can sense the temperature gradient up to 0.014 degrees centigrade in a 1 millimeter distance. So it can sense it very well. And we are saying the ampulla is 2 degrees centigrade warmer than the isthmus, and it goes from lower temperature to higher temperature. So now it is going to the ampulla. So now it has reached where it should be. Now it needs to find out the egg. Where is the egg? And to do that, the egg play a role here, and the cumulus cell, they secrete progesterone. So once the progesterone is sensed by the sperm, progesterone go to the calcium channels. These calcium channels can only open if there are two conditions met. One is that progesterone is bound to these calcium channels. Second is there is high bicarbonate ion in the sperm. When these two things are there, these channels open and more calcium gets in. More calcium gets in, you have more dynein activation, you have more ATP production. Now the sperm becomes really hyperactive. Okay? And that's what we said, that only the capacitated sperm will have high bicarbonate ion, so only they will get that capability to become very hyperactive in the vicinity of the egg. And that hyperactivity is required for the movement of the sperm through the cumulus, reach to the zona pellucida. The third thing that happens is, we are saying that the cell of the cumulus, they secrete CRISP-1. And there is a gradient of CRISP-1, more toward the zona pellucida, high toward the outside, and only the hyperactive or the capacitated sperm can respond to CRISP-1, and that chemoattractant allow this, it's attracting the sperm to move from outside, reach toward the innermost cells. So this is how it is reaching to the zona pellucida. That is making sense? Or no? Okay, so talk to each other for a minute or so we can be clear.
the crisp one from the inner cumulus
Hmm.
layer?
Yeah, yeah, so that's why you have higher gradient from outside to inside. Anything not clear? Oh, they're good. Okay, now. So we didn't talk about acrosome reaction, we said how that happened. That was part of the capacitation process, right? But now, we are talking about acrosomal reaction. We did already talk about how it happened. And essentially what we want to talk about is how the zona pellucida is recognized, right? And recognition of zona pellucida. Okay. So what happened that you have this is your egg membrane, and surrounding this is your zona pellucida, and surrounding these are your cumulus cells. These are your cumulus cells. And we already did talk about the mechanism by which the acrosome reaction takes place. So now, when the sperm are hyperactive, sperm are moving, all those calcium gets in and everything is happening. So people believe that right here, actually, this is the place right in the cumulus cells where acrosome reaction occurs. Okay, so acrosome reaction occurs in cumulus cells. Okay. Now, but what is the first step? This sperm, which is like this, now this is the acrosome process right here. So let me just draw it a little bit so it's more clear. So this is kind of the shape of the sperm. So let me draw it a little bit better, actually. So this is kind of like this thing, and right here is acrosome vesicle, right? And what was the name of the protein we said which traveled from here to here? Isomo, right? That isomo is here. And that leads to, this thing is going to fuse, and right here the actin is there, right? That actin is going to make microfilament, and what is going to happen is, you are going to have a structure, something like this. Let me draw the same colors. This will be the structure after the acrosome reaction. So this is acrosome reaction, and this is the acrosome process. And right here is isomo. Now, so this thing has happened here at this place. Okay? Now, this acrosome process need to go to the zona pellucida and bind to zona pellucida, right? That's how white line membrane is bound to the acrosome process in the sea urchin, same way here. So on the zona pellucida, you have 4 different types of zona proteins. Those are called ZP1, ZP2, zona protein 1, zona protein 2, zona protein 3, zona protein 4. Okay. Now, the binding, when the sperm is coming, this acrosome process, acrosomal process, whatever you want to call it, binds to ZP2. Binds to ZP2. Now what is going to happen? That you have the acrosome process taking place here, acrosome reaction occur here, it releases all the enzyme, right? What those enzymes do, they essentially remove all the cumulus cell from you. So all the cumulus cells are removed because they have many hydrolytic enzymes, many proteases. They basically separate all those cells. Now what you have is, you have an egg and you have a ZP, and right here is your ZP2. To this ZP2, your acrosome process is bound. Now this binding of zona proteins to the sperm is not that much species specific. In the sea urchin, we are saying highly species specific. Here it is not that much species specific, and I think you asked that question right in the beginning, how you get the mule, horse, and donkey. That is the reason. So that is not a highly species specific. Now, the sperm which have undergone acrosome process, they bind to ZP2. But some sperm may reach late, or they have not undergone acrosome process at all. Those sperm, they bind to, so late arriving sperms bind to ZP3. They bind to ZP3. And these sperm are still intact. Because remember, acrosome reaction, everything depends upon zona cells secreting progesterone, all those things, right? More and more calcium coming out, so, but that is not there. So these sperm, they are arriving late, but they can bind to ZP3, but they have not undergone acrosome reaction, so they cannot do anything, they'll just stay there. But even if they're bound, they can still do, there can be other sperm which have been coming a little late, but they have, capacitation have taken place. How to remove those? There is no fast block to, there is no fast block to polyspermy. However, we have cortical granule reaction, and we know how cortical granule reaction works. We have talked that. So far it's okay? Any confusion here? Okay, now what is happening? Here one more thing I should tell you. Remember, we said these cells create CRISP1, right? But there is when the sperm is moving in the reproductive tract from isthmus to ampulla and then to egg, there is a liquid continuously flowing, right? So the sperm actually travels opposite to the flow of liquid. Sperm travel opposite to the flow of liquid, and that process is called rheotaxis. Opposite to flow. Okay? Now, what is happening? So far it is okay? So we are saying that this sperm is attached to GP2, but there might be other GP2 also, right? So during this process what happens, the cortical reaction takes place, the ovastatin clips off GP2. So if any other sperm which is capacitated, which have the capability to bind the egg, once it is bound by the first egg, then cortical reaction produces ovastatin, that is in the cortical granule, and that basically clip off all of the GP2 protein there. And those GP2 protein which are not attached there, which are outside, they are all removed. So no new sperm can come and bind to GP2. So far it is okay? We have 5 minutes, right? Okay, then what happens? This is okay. Any doubts here? No doubt. Okay, then what happens? Now right here is the egg. This is the egg membrane. So now what has happened that here's the other thing. This is the egg. In the sea urchin, we said that sperm come like this is the acrosomal process and bind like this. In the mammal, this thing does not happen. It actually binds in a lateral way. For example, so this part where the isomo is, that part is going to bind to the sperm membrane, okay? And this part is called equatorial region. So right here, the sperm is going to come. This is the acrosomal process. Right here. So here is your isomo on the sperm. Right? So then what is there in the, so remember these are microvilli which are formed on the membrane. What is there in the membrane? In the membrane there is a protein. That protein is called, right here in the membrane there is a protein, that protein is called Zuno. Okay. So isono combined to Zuno. And once that happens, Zuno, both they recruit CD9 and CD9-8 membrane fusion. So membrane fusion takes place. Now one more thing happens. So Zuno is here, Zuno can be here, Zuno can be here, Zuno can be everywhere, right? So say by chance one more sperm gets in. But what happened, once first isomo bind to Zuno, all these Zunos which are here, they move inside in the cytosol. Okay. So that is also preventing polysperm, because there is no way the isomo can bind to Zuno because there is no Zuno. So once first sperm bind, isono bind to Zuno, after that all the Zuno which were present on the membrane, they move inside the cytosol. So what we are saying that sperm binding, sperm movement is eutaxis. That eutaxis is against the gradient of the flow of the liquid. And we are saying there is equatorial region where you have the ZOMO. ZOMO can bind to, isomo can bind to ZUNO. Once it binds, all the ZUNOs are moved in. And before that, we said that binding of sperm to zona pellucida is depending upon the zona protein. So acrosome process bind to ZP2, zona pellucida protein 2. The sperm which are not capacitated, which have not undergone acrosomal reaction, they bind to ZP3. And we said, once the sperm bind to the egg membrane, cortical reaction takes place, ovastatin is released, that will remove all the ZP2, so that no more sperm can bind there. And then sperm bind to the membrane, isomosomo, that recruits CD9, that causes membrane fusion. And then you have all the ZUNO are going in, so no more ZIMO. So in this way, polyspermy is also avoided. So we talk about membrane fusion, how that happened, we talk about how polyspermy is avoided. Any doubt here? Okay, we may have 1 minute, let's talk to each other and say, see, now we have time, we have 3 minutes, right? So talk to each other, make sure that this is clear. Receptors are all proteins. So remember that. Receptors are all proteins. Confusion?
No.
Okay. You're okay?
Yeah.
Actually, we do have a question now.
Hmm?
So, so the cortical granula makes the ovastatin, which comes off the ZP2.
Right.
Does this happen before the late sperm arrive and bind to ZP3, or does do like just one?
So everything is happening side by side. You know, some sperms are coming there, they are not capacitated yet, some are capacitated. If they're capacitated, they need to bind to GP2. So it's kind of like, everything is happening in real time. It's not like one happened, then next happened.
So is it possible
It-
for like two sperm to bind to a Z, like one comes in this way and it binds to
Or that
ZP2,
never
one
happens.
binds to-
No, that never happens. That never happens because this is the way the gradient is. It's not like throughout. That's how the egg adjusts the gradient, and more gradient should be only one side, so they all be
Okay.
on one side.
Okay.
Yeah. But you can have many sperm coming late and binding to many places, but that's the purpose of the next check. When you're removing the ZP2, it means whatever is bound to ZP2 is going to go away. Will have no chance to get in, right? You need to clip. So if you clip ZP2, then the sperm attached to ZP2 is also gone. Making sense? So say, for example, you're saying, okay, here's some sperm come and bind. Okay, it binds, that's okay, but this sperm has already activated the CG reaction. So it's going to, say, multiple steps were happening. Fertilization envelope, that is going to make it away. Then you're having ZP2 being clipped up, ZP2 is gone. And even if it goes in, it will not find any Zuno there because Zuno will be in.
Is there, maybe you know the function of the before?
We don't know.
You know,
We don't know. Yeah, that's what I said. There's so many things don't know, but many things we don't. Okay, so we'll meet tomorrow in the lab. Okay?
When do you think we'll finish this topic? Because this is the last